Hello friends, we are going to talk about the details of the reactions of glycolysis. And the first reaction of glycolysis is the conversion of glucose to glucose 6-phosphate. You can see here, this is glucose molecule and this is converted into glucose 6-phosphate. Phosphate group is attached. Before discussing the chemistry of this reaction, we would see few important key points here, okay, that you would see in the reaction. The first one is the enzyme. What is the enzyme here? This is hexokinase. Enzyme is hexokinase. Hexo means 6 carbon containing sugar, and that is the glucose, and the enzyme is kinase. So the enzyme acting on the sugar is glucose and the enzyme here is kinase, hexokinase. So what is kinase enzyme? Kinase is the enzyme that catalyzes the transfer of terminal phosphoryl groups. Okay, so the enzyme that catalyzes the transfer of terminal phosphoryl groups are called as kinases. So enzyme is clear. Now the requirements of this enzyme. What are the requirements? Requirement means what do we need to catalyze the reaction. The requirements are ATP and Mg2 positive. You can see here ATP is needed and Mg2 positive also needed. And the nature of the enzyme. What kind of reaction is it? What kind of reaction this is? This is irreversible. So this is irreversible reaction. Once the product is formed, it would not back to form the same molecule, that is glucose. So this is irreversible, and also this is androgenic, anabolic you can say, or you can say endothermic, because the energy is needed and it is in the form of ATP. So this is also done. Then the reaction type. What's occurring in the reaction? Phosphorylation. So phosphorylation means there is addition of phosphoryl group on the 6th carbon containing hydroxyl group. So this is about the specifications or the key points of this reaction. Now we are going into the deep detail of this reaction, and in that reaction we are going to see why Mg2 positive is needed, and there is something more, that is why phosphorylation, why we need to phosphorylate, or what is the importance of phosphorylation, significance of phosphorylation, why do we phosphorylate glucose in the beginning, or what is the importance or significance of phosphorylated intermediates. There is a separate video after this video on all these questions. Okay, now we are going into the detail of this reaction. So we have glucose molecule. To save time, I am not drawing all these hydroxyl group here. So this is the 6 carbon where we are interested, and we have ATP. ATP is adenosine. This is nucleoside, and nucleoside which is containing adenine, the base, and the ribose. I would not draw that structure. I would draw only the phosphate groups. 3 phosphates are there. Phosphoryl groups are 3. Okay, so this is negative charges. This is the alpha, beta, and gamma. Now this carbon which is containing the nucleophile, okay, wants to attack on this gamma carbon, but it cannot do, ये कर नहीं पा रहा. What is the reason? What do we mean by nucleophile? First we need to answer this. Nucleophile means something that loves positive charge, but there is negativity, here negative, here negative, here negative, here negative. So how it is going to attack here and take it out, take this terminal phosphoryl group from this ATP. So that is aided by this Mg2 positive. How? We are going to discuss that. This Mg2 positive chelates this. It makes complex with ATP, and in the process what it does, it shields the negative charge and makes the attack of this hydroxyl group nucleophile easy. So now it can attack and remove this. It's that simple. So so we can say the actual substrate for hexokinase is not ATP. ATP is not the actual substrate, but it is complexed ATP with Mg2 positive. 2 positive, 4 negative, so it would become 2 negative. So this is the real substrate. So now it has attacked here. What would be the result? Result is, I remove this first, erase. It would make phosphorylated glucose. It is phosphorylated to this way. So this is phosphorylation of glucose. So this is our glucose 6-phosphate. Now there is something more to know about the binding of glucose and ATP to the enzyme. So there would be some conformational changes in the enzyme when it binds to glucose and ATP. So there are two scenarios. First is when it binds to glucose, and the second when it binds to ATP. So now we are going to discuss that when it binds to glucose, what happens? It is that when it is bound to glucose, or when the enzyme binds to glucose, there is a conformational change. Or simply, the shape of enzyme is changed, and that change is we call induced fit. So that when the enzyme binds to glucose, there is a change in the conformation of the enzyme, that is the induced fit. We are not going into the detail of this induced fit because this is a part of enzymology. For now you remember induced fit change. Okay, now when it binds to ATP, when the enzyme binds to ATP, you can see there are two domains. The one is the red and the other is the green, this blue. So this is U-shaped, you can say U-shaped molecule. Two domains are there. When the ATP is binding to these domains, to this enzyme, it brings these two domains closer. ये दोनों domains को नज़दीक लेकर आता है. How much? By 8 angstrom. So binding of ATP molecule is resulting in bringing these two domains closer by 8 angstrom. Now this binding and this movement of 8 angstrom moving this like 8 angstrom results in bringing this glucose molecule closer to ATP, which is also bound to enzyme. And this you can say this complex blocks the access of water. So this arrangement I can say, this arrangement blocks the access of water. So the water which is present in the solvent can't interfere. It cannot access what's happening here. If it was not like this, that is, if it was not binding to these molecules, that is ATP and the glucose, what could be the result? Result could have been that the phosphoanhydrous bonds which are present in ATP could have been hydrolyzed. Hydrolysis of phosphoanhydride bonds if there was not this kind of arrangement. I think this would help. Thank you.